Welcome to today's episode at Autism is Biomedical. My name is Dr. Christian Bogner, and we have a truly exceptional guest with us today, Stephanie Seneff. Hello, Stephanie.
Hi, happy to be here.
Thank you for your time. I know you're busy in your work. Um, Dr. Seneff embodies the spirit of a true scientist. She explores areas where others may not. She asks the uncomfortable questions, and she looks above and beyond to study how environmental toxins impact our health. Stephanie's groundbreaking research sheds light on complex interactions that many overlook. She's always been available for us to clarify concepts over the years. She always is ready to discuss mechanisms and proposals in regards to autism that I had over the years. And I think both of us were pretty close last year, I think, of writing a paper on a specific subject. Maybe that will still happen. But we are very honored to have you here with us today. And, especially wanted to thank you, especially for your invaluable contributions to science, for our understanding of a big puzzle piece. I think that many of us are not truly aware of yet of how it really works. And it's not simple when you read through the work that you have done. But yeah, historically, for those that don't know, I wanted to mention this because in other podcasts, I don't think it's mentioned enough in regards to your credentials. You're a senior research scientist at MIT Computer Science and Artificial Intelligence Laboratory. You received your Bachelor's of Science degree in Biophysics in 1968. You received a Master's of Science and other degrees in electrical engineering in the 80s, and your PhD degree in electrical engineering and computer science in 1985, all from MIT. And so for the last 3 decades, your research interests have always been at the intersection of biology and computation, developing a computational model for the human auditory system, understanding human language, so as to develop algorithms and systems for human-computer interactions, as well as applying natural language processing techniques to gene predictions. You have published over 170 articles on these subjects, and you have been invited to give keynote speeches at several international conferences, and you have supervised numerous master's and PhD theses at MIT. In the recent years, though, Dr. Seneff has focused her research interests back towards biology. She's concentrating mainly on the relationship between nutrition and health. Since 2011, she has published over 30 papers together with colleagues in various peer-reviewed medical and health-related journals and topics such as modern diseases, Alzheimer's, autism, cardiovascular diseases, analysis and search of databases of drug side effects using NLP techniques, and the impact of nutritional deficiencies and environmental toxins on human health. So, Stephanie, we've known about you and your work in our community for at least for me, for over a decade now. I don't remember, but we have met on multiple occasions in person at different conferences. I remember a dinner with Dr. Mikovits and
Yes.
many years back. So you are a senior research scientist at MIT, and primarily you're known to most of us in the community for your research on the effects of glyphosate, which is
Yes.
the active ingredient in Roundup on human health. You've authored several publications on this topic, notably your book, Toxic Legacy, How the Weedkiller Glyphosate is Destroying. Beautiful, and we'll post a link for that. How the Weedkiller Glyphosate is Destroying Our Health and the Environment. And you've also published various articles and studies exploring glyphosate's impact on biological systems and its potential links to various diseases. Your interest specifically, besides other diseases, is autism, however. And that's how I got to know you, meeting you at Autism One, I think it was, the conference. And your research papers that you have written and published discuss glyphosate's role as an endocrine disruptor, its correlation with rising disease trends, and its biochemical impacts. And we will show the links to your publications and your book during this post-interview editing. But interestingly, you have explored how environmental toxins like glyphosate can disrupt sulfate homeostasis, potentially impacting specifically this molecule called heparin sulfate and related biological processes. And we wanted to focus somewhat on this topic today, the sulfation mechanisms. And we see a lot of benefits in the community, especially as we've discussed previously with the thiosulfate. So now, Stephanie, you believe maybe a better word would be, you know that glyphosate is a significant factor in many modern health crises, including autism, various cancers, GI disorders. And you demonstrate through your work that glyphosate's presence in the environment and food supply is a major public health issue, exacerbated by the ability to disrupt essential biological processes. So let's start from the beginning. What is glyphosate? Since when does it up in my Cheerios? And what are all possible sources of this chemical in our environment?
Right. So glyphosate is the active ingredient in the herbicide Roundup. Most people know about Roundup. Many people use it to kill the dandelions in their yard. Considered to be a wonderful herbicide, it kills all plants except for those that have been engineered to resist it through GMO technology. And it is completely harmless to humans. That's what the message we're given by the government. The government thinks it's harmless to humans. And there's increasing evidence, and especially in recent years, that that's not true. You know, the science is coming out fast and furiously right now. I'm very impressed. Finally, people are realizing it's not safe. And then people who do toxicology studies are choosing to study glyphosate, whereas before it was like, why would I waste money on something that's safe? So they wouldn't bother. And of course, the industry tries to keep people from studying it because they certainly don't want us to know how toxic it is. I think it's far more toxic than we've been led to believe. And unfortunately, it's all over the food supply. It's by far the most common toxic chemical in the food. And I can say that with confidence because Zen Honeycutt, who's a friend of mine, a dear friend of mine, and she's the founder of the organization called Moms Across America. She's been on a mission testing all kinds of different food types, things like fast food and school lunches and even formula and breast milk, all kinds of things she's been testing for glyphosate, and she's been finding it systemically in the system. 100%
Didn't they check vaccines too? MMR, highest concentration?
Yes, that's right. She's the one who tested. There were only two people I know of who tested the vaccines and MMR. I was really
now, they would argue, though, Stephanie, they would argue to say, oh, it's just a couple parts per million. What do you call those arguments?
It was actually a small amount. It was in parts per billion, but glyphosate is extremely toxic. It, at fractions of a part per billion, it's been shown to accelerate the growth of breast cancer cells grown in culture.
So it's actually it's a genius toxin if you would think.
It is.
Or if you want to introduce something into the food that kind of doesn't kill you, you don't really feel it. It's kind of a longer process for you to get sick chronically. And I remember when I was working in the OBGYN world, we had these samples for prenatal vitamins. I did send one of those because the main ingredient was soy, GMO. It didn't say non-GMO. Yes. So I sent it in, and sure enough, it also came back. And I remember I published that on my blog, but it didn't get any attention. But I stopped giving. I don't want to mention the brand, but you could guess which one. Contained glyphosate. We're giving that to pregnant patients. So if you tell me,
that's something that I've really become aware of. And in fact, I even wrote a paper on drug side effects possibly related to glyphosate in the drug, and especially drugs that are injected. It's a very serious problem, I think, with when you inject glyphosate. And that's what you do with the vaccines. You get 100% of it into the system, into the blood, really. It's past the barriers. The gut can help to keep glyphosate contained to some extent.
We have thousands of different toxins in our food, right? Many of them, I think, not even studied properly by the EPA to see, human safety data. But what made you focus? I know you focus on other chemicals too, but what made you specifically? What sparked your interest in glyphosate?
And you're right. I've totally zeroed in on glyphosate. I was looking for all kinds of things. I started getting very interested in autism around 2007. And that's actually when I switched my career path away from, I was building computational models for human-computer interaction. I was building systems that would allow computers to talk to humans in conversations like Siri and Amazon Echo. So our systems were precursors to that technology. But I switched over completely to biology in 2007, specifically because I saw the autism rates going up, and I was very worried. And I thought the government thinks we're just diagnosing it more, and that's not true. I just really was sure it wasn't true, and I wanted to find out what is it in the environment that's causing autism. And I looked for 5 years, and I looked at lots of things. And I think the vaccines are a factor, and I certainly considered the vaccines, but I still felt after 5 years I hadn't found the true cause. But I knew there was something about the gut, because a lot of the autistic kids have a lot of gut problems. They have food sensitivities, they have diarrhea and constipation and bloating. I mean, all kinds of issues with their gut, food sensitivities, food issues. They're not good eaters, very picky eaters. Typically, many of the autistic kids are like that. And so I figured it had to be something in the food. I had zeroed in on the idea that it must be something in the food, but I didn't know what. And I didn't consider glyphosate at all, because glyphosate's safe. Why consider glyphosate, right?
Right.
And I was very fortunate to happen to be at a conference where Professor Don Huber was giving a 2-hour presentation on glyphosate. That's the first that I had really heard of glyphosate. I mean, I knew about Roundup, but I didn't pay any attention to it, right? Because it's safe. That's the thing. People don't think of it because it's safe. And that's a huge, huge problem. We think it's safe. Therefore, we're not worried about it. Even the regulators aren't worried about it. Nobody's policing it. And then it's such a great boon in agriculture, because it kills the weeds without having to kill the plants, because you can GMO engineer the plants to be resistant. So you can spray it all over the crop. It doesn't die, but it does soak it up. And so it gets into your food supply. And, Zen did a recent study, and she looked at all the different, lots and lots of different chemicals in agriculture. And glyphosate was the only one that was 100, basically 100%. I think there were 2 organic. She had 46 items and 2 organics. These were gluten-free foods. 2 out of 46 were negative for glyphosate, and they were both organic. So if it's not organic, basically it's got glyphosate. I mean, that's almost a rule of thumb, especially processed foods. She found that 100% in the fast food samples, 95% in the school lunch samples. So she's been really showing that it is pervasive. Whereas the next highest one was 2,4-D, which is a serious chemical, and that was found in like a third, I think, of the samples in this last round. But she tested all kinds of things, and most of them are not there. It's really glyphosate is dominant, and we were sort of tied with Brazil for using the most per person in agriculture. U.S. is in a race against Brazil as to who's going to be the sickest, basically.
I was for about 3 years. There was a physician on the East Coast that, together with Great Plains Laboratory, they had this mold toxin test for the urine. I did that for several years, and during my studies there, to figure out why there's so many different mold toxins in the urine, I came to the conclusion, or I found that the US, along with India and China, have the worst crop mold contamination on the planet.
Interesting.
And that they are. Do you think that the glyphosate played a role in the crop's immune system to maybe? I mean, a better question. The principal mechanisms. Why do they spray this on crops? There's the shikimate pathway. Can you maybe expand on that? And
sure. Well, I'll start with the mold, because Aspergillus, and I wrote about this in my book, is a serious mold problem, like in the corn and things like that. Aspergillus is one of the very few microbes that's able to metabolize glyphosate, and that gives them a huge advantage. So that it kills off a lot of bacteria, and the mold gets a huge advantage because it's able to metabolize the glyphosate and actually clear it, which is doing a great service to the host. It does. I think it does it for us as well. Aspergillus is it's a blessing, but it also a curse because it's so toxic. And the same thing with Pseudomonas aeruginosa. That's a bacterium that can break down glyphosate. And we've had a huge problem with Pseudomonas in the hospitals lately, with infections with Pseudomonas.
Wow. Well, interesting link there, because as you know, there are 2 bacteria that we discovered that can produce cyanides, which are Prevotella copri and Pseudomonas aeruginosa.
Interesting. We could get right to the cyanide, because that's very, very interesting. And you just introduced it.
Maybe not yet. Maybe not yet.
Yeah, I might be rushing ahead.
Explain the shikimate pathway that they inhibit, and why do they tell us that it doesn't affect us? Do we not have that way?
Yeah, that's actually the bottom, the really easy message that sounds very reassuring, which is to say it affects this particular enzyme EPSB synthase, which is an enzyme in the shikimate pathway, which is a very important biological pathway for the plants and also for the bugs, for the microbes. But human cells don't even have that pathway. We're relying on other folks to do that part for us. It produces essential nutrients for the host through this pathway. And glyphosate blocks it. And so this ends up with deficiencies in the products of that pathway, because the microbes are producing those products for us using that pathway. That's the flaw in the argument. They say, well, human cells don't have that pathway. So yay, we're not sensitive to glyphosate.
There was a very important point that you just made. So they're telling us we don't have that pathway, but you're telling us our bacteria do have that pathway. Okay, so there we go. So glyphosate pretty much affects our microbiome. What have you found out in regards to the direct effects on the microbiome with glyphosate exposure in our diet?
A very important part. And I actually wrote a chapter on the gut in my book, and I worked really, really hard on that chapter. I had to read a lot of stuff and sort through. It's hard, because there's a lot of papers coming out, and they've got these pretty charts with all these colors, and all these different bacteria and different amounts of this and that. And it's just like your eyes glaze over. You just can't process it. So it takes quite a while to sort of sort through that and figure out exactly what it is at the highest level, what's going on. And I am proud of myself for figuring that out. And I wrote about it in that chapter, and I believe I'm right. And it has to do, of course, with glyphosate. The beneficial bacteria are more sensitive to glyphosate than the pathogens. And so what happens is the glyphosate kills them off. And that's specifically Lactobacillus and Bifido bacteria. Those two are dominant in the infant gut. And glyphosate, they're both very sensitive to glyphosate. And then they have the shikimate pathway, and they use it to produce these aromatic amino acids, which are essential to us because our cells can't make those. We don't have the pathway, we can't make them. So we depend upon the microbes to help to supply us with those critical nutrients. And the bigger problem is really complicated because the lactobacillus help us break down gluten and casein. These are proteins that have a lot of proline. Proline is an amino acid that's unusual, and it requires special enzymes to break it down. And again, we're deficient in certain enzymes that are needed to break the proline apart from the rest of the protein. And the lactobacillus provide those enzymes, but they get killed by the glyphosate. And then those gluten and casein don't get properly broken down. So we have these peptides that stick around with proline in them, and they become allergenic. That's when you get the immune cells are upset to see those. Glyphosate sets up a leaky gut, so those peptides start getting into the circulation. And the immune cells are upset to see these foreign peptides, and so they develop antibodies. But unfortunately, they have similarities to human proteins that are very important. So you end up with thyroid, hypothyroid, thyroids.
Or antibodies against brain structures.
Right, antibodies against various proteins that are really important for the brain. And hypothyroidism is one of them that's like commonly associated with gluten intolerance or celiac disease. So I think the whole epidemic that we're seeing, and we have all these sections of the grocery store with lots of gluten-free foods, that was never true for most of my life. I started seeing it maybe 10 years ago, and I was puzzled. Why do we have all this gluten-free stuff? And then I figured out because the wheat is actually non-GMO. So you think it's safe, but they spray the wheat with glyphosate.
Right, anyway, to keep it dry, right? Yeah. And, for those that are listening, and read between the lines, when you just said that Lactobacillus and Bifidobacterium are targeted by glyphosate, and as you know, our colleague Alex, who analyzes the 16S microbiome sequencing of bacteria, has written his own software to kind of quantify the distribution of these different bacteria. I think most reports that I read come back with low lactobacillus and low bifidobacterium. Yes. And of course, with the urine analysis that we do with certain labs, the glyphosate is often always, elevated. We don't know what the cutoff is, but it's there. It's detected. So it's in the urine, yes. So it's in your system. So it's very interesting. Now, you previously mentioned, and I really wanted to dive into that a little more. You mentioned heparin sulfate, and I had some questions about this. You noted in your research that heparin sulfate is very prevalent and active in the ventricles in the brain, and that it is involved in neurodevelopment, neuroprotection, synaptic function and plasticity, regulation of signaling molecules, the blood-brain barrier integrity, and of course, neuroinflammation. And you also mentioned that it's involved in adhesions of white blood cells to the inflamed endothelium, ultimately leading to chronic neuroinflammation that we actually see in autism. And you linked it to autism, but looking at this from a 40,000-foot view, you basically, from what I gathered, and please correct me, you proposed that glyphosate causes melatonin suppression, glutamate neuroexcitotoxicity, and other factors leading to a suppression of what you mentioned in your most recent paper, a PIN1 activity inhibition, initiating a cascade of events leading to autism.
That's right.
The symptoms that we observe in those that they label autistic. So in a recent interview with Dr. Brandon McLaughlin, which I thought was fascinating, you mentioned that heparin sulfate interferes with transferases in the ventricles, that there's a deficiency in this enzyme putting on the sulfate on heparin sulfate and heparin sulfate itself. My question is, can you explain this specific concept, the transferase deficiency or toxicity? Is this blocked by glyphosate, or are there any genetic reasons that interfere in building it? And how does your latest research into the PIN1 gene link into all of this?
Yeah, okay. You can see there's a lot of dots to connect. And I love a puzzle. And I'll tell you, this biology is such a thrill ride because it's so difficult to sort through and figure out in all the noise, what is the thing I should focus in on and what should I dive deep? Where should I dive deep? That's always the question that I ask. And I have things coming my way all the time that, oh, I should follow this, but oh my God, I'm busy with these other things, so I won't right now. And then I realize that I should have grabbed that. So I'm constantly on the lookout for new surprises.
The melatonin angle was just blown away. No wonder that when we give melatonin, it doesn't work for many kids. And can you maybe elaborate at least on that? Because we know our kids don't like to sleep. If they sleep, they don't sleep deep enough. They don't reach that stage 5 where they dream, where we detoxify the brain. Can you explain the mechanisms behind that? Like, how does this
Now that gets really complicated, but yeah, a lot of these nutritional supplements, I think that there's a flaw in the supplements in that many of them are synthetic. They're made in the chemistry lab. They're not made naturally. And you would think it'd be the same thing, if you make it in the chemistry lab versus you let a biological organism make it. They're not the same thing. And this is something I've really gotten interested in lately. So methionine is a good example, and methionine is an essential amino acid. It's essential because the gut microbes, again, make it for the host, and glyphosate suppresses the synthesis of methionine by the gut microbes. And so we end up with a methionine deficiency. There's a whole weird study area around the idea that methionine is bad for you, and that if you eat a methionine deficient diet, you'll live longer. I don't know if you've heard that rumor. I think it's really, really wild. That you should eat a methionine deficient diet.
Just like fat is bad, and then they put sugar in the yogurt.
It's really fascinating. And that was based on a study on rats, and I looked into it. I'll have to just get into this deuterium thing, because deuterium is what I'm obsessed with lately. Deuterium is heavy hydrogen. And it's bad for the mitochondria. And the mitochondrial deficiency is associated with a huge list of chronic diseases. Just about everything is associated with mitochondrial deficiency, which is due to, I believe, too much deuterium in the mitochondria. Deuterium is heavy hydrogen. It's got an extra neutrons, twice as heavy as regular hydrogen. It's a natural element. It's all over the place. You can't avoid it. But the whole biological framework of metabolism is obsessed with keeping deuterium out of mitochondria. Really, really interesting. And glyphosate is a train wreck for that whole system. This is what I'm really, really interested in lately. And I've been really, again, fascinated with the science and extremely disturbed by what I'm seeing. So methionine, it's the universal methyl donor. You probably are aware of that. And methylation pathways are often disrupted in autism. Both sulfation and methylation pathways are disrupted in autism. And both are probably equally problematic for the autistic kids. And methyl is CH3. And the methionine carries a methyl attached to its sulfur atom, CH3. And if you trace that methyl back to where it came from, the gut microbes, it's going to be extremely depleted in deuterium. That methyl is going to be pure hydrogen, no deuterium, because of the way it's built. And that has to do with hydrogen gas that's produced by the gut microbes. And when you produce hydrogen gas, the gas loses 80% of the deuterium. And then you put that gas back into organic matter to make methane. Methane is a gas that escapes from the gut. Microbes are making it, and they're making it from hydrogen gas, and they made that hydrogen gas from organic molecules. But they did that on purpose to get rid of the deuterium. It's really, really fascinating stuff. It's wild, wild science. I'm super excited about it. I can't tell you enough how excited I am.
When the deuterium enters the brain, what is the sequence of events, in your opinion, in regards to how you link also your PIN1 gene research and the sulfate?
Yeah, well, I get off on tangents, but the sulfate is super important. Heparin sulfate in particular, which we talked about. Heparin sulfate, it decorates all the blood vessels. It's the extracellular matrix, that surrounds all the cells, and then it lines all the blood vessels. The endothelial cells that line the blood vessels have all this heparin sulfate stuck to them. And the heparin sulfate has sulfate, and the sulfate gels water. And so what that does is it traps gelled water around the blood vessel, creating a barrier. It's called exclusion zone water. And this is Gerald Pollack's work. That's another one that I was really fascinated with. I've known Gerald for many years now, and he led me down this path of the heparin sulfate, the gelled water, and it creates a battery, and the battery pushes protons out into the blood. And so the gel becomes negatively charged. But those protons that go out are not deuterons. This is the really important thing, is that it traps the deuterons. So
Stephanie?
The deuterium.
Oh, the deuterium.
Yeah, it traps it in the heparin sulfate in the water. It's so cool. It leaves behind the deuterium because the hydrogen is much more readily able to leave the gel than the deuterium. So it's sort of a fractionation process that goes on.
But that's not a good thing, right? That the deuterium.
It's a good thing because you're trapping the deuterium in the gel, which is keeping it well away from the mitochondria. You're keeping it sequestered.
I thought it would get
absorbed in the gel
and then be toxic.
And you end up dumping out protons, which are not deuterons, into the blood. So the blood gets low deuterium. The whole system is geared around where can we put the deuterium to keep it away from the mitochondria. That's the entire system of metabolism. It's
but if it traps it, where does it go?
It stays in the gel. So you have the deuterium in your body, but it's sequestered in certain places and kept out of certain other places. So in the cytoplasm, the cell doesn't care that much about deuterium. I mean, it's not high, but it doesn't have to be excessively low in the cytoplasm because that's not where the mitochondria are. But you have these organelles, and it includes the lysosomes and the ER, the endoplasmic reticulum, and the nucleus and the mitochondria, all of those organelles that are inside the cell. I bet you all of them have low deuterium. And they all coordinate with each other, and they share their water. And the mitochondria make water. They're making lots and lots of water all the time. Whenever they make an ATP molecule, they're making water. And that water is deuterium depleted. It's called metabolic water. And it's going to be very low in deuterium because they have pushed into the intramembrane space all these protons, and they just hate to put deuterium in there. They always want to make sure it's a proton. And they do that through very tricky enzymes that are specialized in avoiding deuterium. It's extremely fascinating stuff. And it has to do with starting with those molecules that are made by the gut microbes from the hydrogen gas. So you go all the way back to the hydrogen gas. And of course, hydrogen has become a therapy that's quite popular, I think, even with autistic kids. A lot of people are kind of into hydrogen therapy. And it sounds a little bogus. And when I first heard about it, I was like, I don't think so. It sounds a little strange, But now I realize that it is actually valid, hydrogen therapy. And also you can get deuterium-depleted water. They call it light water. And you can drink that as a way to get low deuterium water.
So as we know that in autism, I think previously mentioned, we have higher excretions of sulfates found in autistic urine, including thiosulfate and metabolite. So if we have sulfur deficiency or sulfation, sulfation pathway interruption in autism, what are the effects of the deuterium then in those systems on the mitochondria?
Yeah, well, again, everything is quite complicated. But the thing is, if you can't put sulfate onto things, which is what happens, all of these they're called sulfotransferases. And that was the thing that I got interested in the idea that sulfotransferases were deficient in autism very early on. I was looking at Rosemary Waring's work, and I don't know if you're aware of Rosemary Waring. She went way back into the 1990s. She treated autistic kids. She had a lot of them that she dealt with, and she measured metabolites in their urine, and she found out they had incredibly high levels of sulfide and thiosulfate in their urine, way out of line. And so she suspected they had some kind of problem with the sulfation pathway, and she proposed, in fact, a deficiency in sulfotransferases, which was absolutely brilliant. And this was, even before I knew about glyphosate, I knew about her work. And I was kind of focusing in on this problem with the sulfation pathways. I suspected that was a crucial aspect of autism. And I definitely believe that now. I'm quite confident that that's true. And the problem is because glyphosate just suppresses sulfotransferases. And this is theoretical because I can show it with respect to, in my book, I talk about a glyphosate susceptibility motif in proteins. And I suspect highly that glyphosate is getting into proteins by mistake in place of the coding amino acid glycine. It is a glycine molecule. It has extra material stuck onto its nitrogen atom, but it is a glycine molecule. So it can fool the system. It's an amino acid. Glyphosate is an amino acid. And so what happens, I believe, is the protein ends up putting glyphosate in where it should be putting glycine. And that can be catastrophic for certain proteins. And so in particular
Such as
proteins that,
glutathione, huh?
yeah, glutathione has a glycine. So that's going to be problematic. And actually, I think that some of the trouble with glutathione that we're seeing with autistic kids has to do with glyphosate substituting for glycine in the glutathione molecule. And that's going to make it behave very badly. It's not going to do its job. So, but the sulfotransferases, they have a motif. There's a characteristic motif. It's called a GXXGXXG motif. G stands for glycine. So it's a sequence of amino acids. X could be any amino acid, but in between. But there's 3 glycines that have to be there in all these different sulfotransferases. They all have it, different species, different roles, like they can transfer sulfate to different things. They all have it. And that motif, and it's at the site where they bind phosphate. So it's actually exactly what I call the glyphosate susceptibility motif. That defines it. And these guys have it, all these sulfotransferases. And so there was a recent paper on autism, very last couple of years, where they found specifically that there were deficiencies in phenol sulfotransferase in the gut and phenol sulfotransferase in the blood and heparin sulfotransferase in the brain ventricles, in the pineal gland. They actually did that post-mortem on the autistic kids. The autistic kids had a problem with sulfotransferases. It was very clear, systemically. And so this is how you get deficiencies in heparin sulfate. And you need to attach sulfate to things in order to transport it, because if you just let it run free, free sulfate in the blood will gel the blood. You can't do that. So there's a real problem with sulfate transfer. They're trying to get the sulfate all over the body to get into those heparin sulfate proteoglycans, but if they can't attach it to the heparin sulfate, then they're screwed, basically. They're going to have 2 problems. One is the blood is getting gelled, and 2 is the heparin sulfate's not being formed, so there's not enough gel lining blood vessels, and you've got a complete disaster in terms of maintaining that deuterium system that works so beautifully when you don't have that problem.
So it's fascinating, and I know that you mentioned specifically the defect in the heparin sulfate not being able then to basically attach sulfur to melatonin, right, to make it really work.
Well, it's melatonin sulfate is another one. So melatonin synthesis gets suppressed in the pineal gland by glyphosate. There's papers that show that. There was a paper that showed something like 45% reduction in melatonin synthesis in the pineal gland in a rat study. Melatonin was suppressed, and it's not surprising because melatonin comes out of the shikimate pathway. You have the 3 aromatic amino acids, and the tryptophan, tyrosine, and phenylalanine, and the tyrosine is a precursor to melatonin. So when you don't have enough tyrosine, you don't have enough melatonin. In fact, I think the black boys may be more susceptible to glyphosate simply because they have dark skin. They need to make melanin, and that's going to draw away resources to make things like dopamine and melatonin and serotonin in the brain. So they get a greater deficiency in those critical neurotransmitters as a consequence of having to make all the melanin that's in their skin, which is competing for the same resources that are deficient.
Wow. As you know, we've talked about this before, and I don't want to dive too deep into that, but we have looked into naturally occurring cyanides and also the cyanides that we discussed, the Pseudomonas and the Prevotella, that may be produced in excess if there's dysbiosis. And the way this is broken down in the body is by sulfotransferase called rhodinase, which is expressed in the gut, liver, brain, kidneys. And what do you think about that? Could there be other sulfotransferases that are blocked by glyphosate and with this mechanism that you explained?
I have to say that I saw that, you sent me that email right before we got on, and I was like, oh my God, rhodinase. I wonder, it's one of those things I've run across, and never quite connected. I didn't quite realize that it was a sulfotransferase. And then I just looked at it just before I got on with you, and I'm super excited about it because I think I need to dive into it. I think it's going to turn out to be a critical piece of the puzzle for the autistic kids. That is a sulfotransferase, and it detoxes a cyanide. Mm-hmm. And I suspect that it's getting suppressed by glyphosate. I haven't quite looked at it to see if it has that motif, but I'll bet it does. And because I know how to find that out. But
disulfide is used in the ERs, right, for cyanide toxicity. But if this enzyme doesn't work, you won't break down the cyanide to thiocyanide. And it's, I think, very critical, not just for autism, but just in general, for
I
all.
think so too. I think you're onto something big, and we could write a paper on that. You and I could write a paper on that
That's
together,
what we
I
wanted to do.
suspect.
We should.
Yes.
So in regards to, you mentioned in an email previously that you may have some recommendations, and maybe I've mentioned them already in a previous interviews, but what can we do? We have the problem. It's too late for many of us. Our children are not speaking, they're autistic, they have this going on. What would you suggest we can do? Are there any natural remedies?
Well, certainly organic diet. And fortunately, in the United States, we do have an organic label. It's not bad. They can't use glyphosate. If they do, it's illegal. So you cannot use glyphosate on the organic crops. Unfortunately, it can still be there because it can be in the manure, for example. They don't have to have organic manure. And the cows eat a lot of glyphosate. So you can end up with glyphosate on the crop, even though you're not using it. But generally, the organics test much lower and often negative on glyphosate compared to the other foods. And so eating certified organic, I think, will help a lot to reduce your glyphosate burden. If you suspect, like if you're living in a farming community, you want to make sure your water is not contaminated with glyphosate. You can test it. And if it is, you can install a reverse osmosis filter that can take it out. And I would recommend doing that. And so
those particular bacteria in the microbiome that you have come across that are able to metabolize.
I mentioned the mold and the Pseudomonas aeruginosa. Those are both not a good idea. So you would, I suspect some people actually have infections that are causing them a lot of trouble. And that if they take antibiotics, they kill off those bugs, and then they have a glyphosate problem. So you sort of are stuck. You're caught between a rock and a hard place, actually, when you have a situation like that. But there are also Acetobacter. These are the only ones I know of that's pretty much my whole list of microbes. It's a tough molecule to break down, but Acetobacter, some of the Acetobacter species can break it down.
So you're basically suggesting also that the initial kill off or the initial toxicity of glyphosate on bacteria like Lactobacillus and Bifidobacterium, that suppression causes an overgrowth of bacteria, for example, Pseudomonas, which then, if you have glyphosate, it's much more potentiated than somebody would have a more balanced microbiome. Is
absolutely? Glyphosate's killing off the other guys, and in fact, another one is the cyanobacteria. The blue-green algae. They call them blue-green algae, but they're actually bacteria. Cyanobacteria are causing a lot of troubles in the Great Lakes. Again, producing toxins that can kill you and stuff like that, and make your seafood not safe to eat. They also can break down glyphosate, and I suspect that's why they thrive. So whenever you are a species that can break it down, you've got a big advantage over everybody else. So you're going to become dominant in that area if glyphosate's poisoning everybody else. You can break it down. You can even use it as a resource. Those cyanobacteria, they probably love glyphosate. You get nitrogen, you get phosphorus. It's a good nutrient in that sense. It's supplying these important nutrients, but of course it's doing it in a horrible way by virtue of all of its problems with the disruptions that it causes in your metabolism. So, but the Acetobacter are found in apple cider vinegar and sauerkraut and kombucha and kimchi, all these kinds of fermented foods. And so I recommend eating fermented foods on a regular basis. I think that can possibly, those Acetobacter can be breaking down the glyphosate enzymatically. And there are some things that can break down glyphosate non-enzymatically. That's through just being very reactive. And those are always tricky because they can also react with other things and cause trouble. But
one interesting thing when you study the chemistry and history of thiosulfate, when you look at the Roundup, kind of the package insert for farmers, they say do not use with thiosulfate.
Oh, interesting.
And because it's, I guess, less effective. So I'm wondering always, does thiosulfate have a binding capacity or deactivating ability of glyphosate? I don't know. Maybe that should be studied too. Now, I know you are heavily involved in research. You've published so much. You're not a clinician. However, when we have a parent with a child with autism, and they're being told that it's neurodevelopmental, there's literally just, it's better diagnosis, it's neurodiversity, whatever, go and have speech therapy, occupational therapy, ABA therapy. What do you think about that?
Well, I'm upset that people aren't recognizing the important nutritional aspects of autism. And I think you can actually get a lot of gain out of just feeding them well. And it really means eating certified organic whole foods, stay away from the packaged foods. And so, and of course, if you can get them to eat fermented foods, that would be great. And eat high fat diet, actually. I think that the fats are actually low deuterium nutrients, and in particular animal fats. So things like oysters and clams and lobster, all those seafood are very nutritious. They have a lot of micronutrients and they have healthy fats. And so you really need to focus on the diet. And I know a lot of the autistic kids are really difficult. They don't like a lot of foods,
Yeah.
which makes it especially difficult. And I think that's also a consequence of glyphosate. I actually found an enzyme. I won't be able to tell you the name. I think it's a dehydrogenase, and it's involved in processing fats. And it has an incredibly profuse glyphosate susceptibility motif. It's got multiple glyphosate susceptibility motifs in this protein. I've not seen anything else like it. And it can be defective genetically. And when it is genetically defective, the child is a picky eater. They can't eat a lot of foods, and they can have a high risk of autism. So it's like kind of the perfect protein to link to autism. And it's a dehydrogenase, which I've identified dehydrogenases as a class, just like the sulfotransferases as a class. They're deficient. They're affected badly by glyphosate, and they're super critical important for deuterium. The dehydrogenases play an incredibly important role in delivering low deuterium protons to the mitochondria. So I think that's another way you connect the dots. It's really the methylation pathways and the sulfation pathways are both disrupted by glyphosate. And a lot of that has to do with these proteins that are susceptible to suppression by glyphosate.
With all of this knowledge that you have, Stephanie, it's like revolutionary, I think. But why do you think, with all of these papers coming out, including yours, and this knowledge and this understanding of this big puzzle, why do you think it's still such a taboo to say autism is medical?
I know, I do not understand that at all. That is so annoying. And I think it has to relate to the industry, the powerful industries that are controlling the regulators. And a lot of that's financial, that they don't want to speak out against the industries that are feeding them money, basically. It's really very corrupt, I think. It's an extremely sad situation.
So what can we do? What can we as parents do other than eating better? How can we bring change or more awareness to this?
And I wanted to mention a couple other things that could be useful. One is deuterium-depleted water, light water. It's expensive. I actually drink it. I have some here right now. I drink it a glass every day with only a third of a cup. So I just put a little bit. It turns this into glacier water. So normal water.
Can you order it online or?
You can. Yeah, and I have a constant supply. I've been drinking it for a long time. Just a little tiny bit every day. It's almost like a supplement, right? And it's the water that you buy is extremely low in deuterium, and you don't want to drink it straight because that's not natural. You just mix it in with regular water. So you only need a third of a cup a day. And it'll give you a chance to get a low deuterium source of water. So people who live in Iceland, for example, are very healthy. And Iceland gets their water from glaciers, and glacier melt is a low deuterium water. So I try to make this be similar to glacier water, what I'm drinking.
Wow. Okay.
And then the other thing is sulfur. Epsom salt baths is something that I'm very fond of. And I do that regularly myself. And I think it would be very good for autistic kids because it's a way to get sulfur into the system by bypassing the gut. And I think there's a lot of issues in the gut. A lot of people have sulfur sensitivity. And again, that's caused by glyphosate because glyphosate disrupts the detoxification of sulfite. Sulfite is very reactive and it can get converted into sulfate, but that enzyme gets suppressed by glyphosate.
SUOX, right? Sulfite oxidase, which is also inhibited by arsenic and tungsten, as far as I know, and stimulated manganese.
Yeah.
That's fascinating. So those are great tips of what we can do. Now, in regards to causing change, though, and awareness, do you think, what do we need? Do we need more studies to show it, or do you think it's
we need people to just jump into action. I think I'm hoping mothers and fathers of kids who are sick, recognizing that the food is toxic, and then there are people who have the stomach for it, who can really fight the regulatory process. You have to have a lot of. It's hard to deal with the regulators because they're really perverse. It's so difficult. I find it very discouraging to deal with them. But those people who can stomach it, I would love to see them And even locally, you can get them to stop using glyphosate on the schoolyards. They did that here in Hawaii. They passed a law recently in the government, no more glyphosate on the schoolyards. And you can, and then you can try to get grocery stores. You can get them to not sell glyphosate. You can do things locally, that can make a difference. And then, of course, when you buy organic food, vote with your pocketbook. And so if enough people are buying organics, the grocery stores will start supplying more organics. They'll see that they're flying off the shelves, and they'll stock more. And that's happening because even our local grocery store is getting more and more organic choices, that we can buy things there instead of going to specialty shops and things like that.
Now, you mentioned Zen Honeycut at Moms Across America. They found it everywhere, right? I found it in prenatal vitamins. You found it during your studies. She found it in vaccines and whatever, your Cheerios, your
breast milk.
Breast milk. Why is nobody listening to this? I don't understand.
She's working really hard. I really admire her, and she's really a doer. I mean, she goes to Washington. She has to get money from donations and fly to Washington and sit with the legislators and present to her results. And it's really frustrating.
Eventually, there will be a block, right, somewhere up.
Yeah, I mean, the government. I was one of people. There were 6 or 7 of us who, on our own nickel, went to Washington and met with EPA about glyphosate, and I had slides prepared and everything, and that was pretty much a bust. I mean, they showed up late, and then they started blabbering stuff that was unimportant and wasting time. And by the time we were ready to present, there was so little time, we had to rush through our presentation, and they acted like we weren't in the room, and they were just like, they were not paying attention. They had no questions. It was just awful. I was so discouraged, and of course, no impact. I mean, nothing happened. So it's a really hard thing to deal with the government. That's why we need to just do this outside of the government. I think we need to tell your neighbors, tell your family, tell your friends, don't eat the regular food, buy organic, and we can set up our own revolution without having to involve the government on that. I think that's going to be the best path.
Well, we certainly have received lots and lots of feedback by interfering with the sulfation pathway, with microbiome rebalancing, with Alex, the work that we have done. And it's very satisfying to see that these theories in the research that you present are translatable into clinical results.
It's very satisfying for me, because I'm just working with the theory, and I sort of have this idea that this must be the thing to do. And then when I hear from somebody who says, yes, I did what you suggested, and now I'm much better, I just feel so gratified.
I have to admit, though, even though working in this space, for the last decade, you write these papers and I'm like, whoa, it's, sulfur, but none of us knew what to do, really. What's the difference between sulfates? Why can't you just give sulfites? Why does a kid react to NAC, and
acetylcysteine, yes.
Yeah, and so it all makes much more sense now, and I really want to thank you, because without this knowledge, we wouldn't be here and have all of these patients being helped. But yeah, I mean, it's fascinating. I can't wait to read some of your future work and to have more discussions. But unfortunately, that brings us to the end of today's lightning episode. We had the privilege of talking to you, Stephanie. You're a true pioneer in exploring the impact of environmental toxins on our health. Your dedication to ask tough questions and your relentless pursuit of understanding complex biological interactions have been truly inspiring. We deeply appreciate your collaborative efforts that you have with us, your groundbreaking research, especially in helping our children. So thank you so much, Stephanie, for joining us and sharing your insights. And it has been an honor to have you.
And thank you so much for doing this. I really appreciate it.
All right.
Important work.
Stephanie, have a great day.
You too. Bye-bye.